Thanks. Yes, hi. So I'm Mario Lestas. I am an associate professor at a small private university in Cyprus. The topic of today's talk is it fits very well with the previous presentation because the previous presentation was all about testbeds for molecular communications. So here is I'm going to present some very preliminary work on a project that was approved for funding lately. So we started 2 months ago. It's very recent, and I'm going to mostly present the idea behind this new testbed that we want to build for validating molecular communications. And also explore some potential applications of this specific system. The motivation behind this was, so a few years ago I started working with a PhD student on molecular communications, and we dealt with modeling, mathematics, but some very preliminary work. But then, the main question was, how do we know that all this is true? How do we validate? And the other big thing is, what are the applications of this? And then I have, one of the co-authors there, Andonis Kirmizis, he's a biologist. So I talked to him about this. So his field of expertise is yeast. So he's an ERC recipient on yeast. So he told me, okay, we can build this system. So this is what I'm going to be presenting today. So it's the first 3 authors. One is a PhD student, the other is a master's student that we did some preliminary work, and the rest are part of the project. So Andonis Kirmizis, he's the biologist, and together with a postdoc, they will be performing the biological experiments. And then we have Andreas Pitsilidis and Konstantinos Piteris from the University of Cyprus. Kostas Piteris, he will be exploring basically the potential applications. Kostas, he's the coordinator of the Fed Open Action Gladiator, which deals with molecular communications. And then we have Ian Kildis, he's a well-known figure in the field, as our consultant. So what is the objective? Well, the objective is what I said earlier. We want to build a new testbed for molecular communications. Right now, and the main thing is, we have seen previous testbeds. I think we wanted to build something that I would say is biocompatible. So we wanted to, the first question is, do cells communicate? Do living organisms communicate, the cells? And if they do communicate, so do we have examples? And then if they do communicate, can we engineer them to perform the communication the way that we want them and not the way that they want? That's the other question. So bacteria, prokaryotic systems have been extensively used. So there have been a few experimental several using bacteria, but yeast has not. Now, why have we chosen yeast? Now, yeast is one thing is that there is it's one of the it's very well studied in the system in biology, in systems biology. So we know a lot about it. Okay, and the other thing is its genetic amenability. We can do a lot of things with yeast. Okay, and this is the main motivation behind trying to build this system. Now, so then I had some slides on previous testbeds and stuff, but Murat has done a very good job in presenting all this stuff. So I will not go into this. So I will go directly to what we want to do. So this is our system. So these are yeast cells. Why do yeast cells communicate? Well, they communicate for mating purposes. Okay, so they are haploids. They have, they call them MAT alpha and MAT A cells. So one is supposedly the male, and the other is supposedly the female. And they, one sends pheromones to each other to initiate, to trigger the mating procedure. Okay? Well, supposedly, each type sends a different kinds of pheromones, not very different. However, we have considered here the, I would call this the MAT alpha cell type, and this is the MAT A. I don't know why biologists they use so close names for this type, but this is what. So this will be made. It does not do that directly. So this will be engineered to secrete pheromones using what we call the galactose pathway in yeast, and then this one, upon receiving, so these are receptors. I will talk about the mechanism a bit more in detail later on. So these will be engineered again to secrete a green fluorescent protein upon the alpha factor reaching this receiver molecule, and then and so the intensity of the light will be used as a measure of the of molecular communications. All right. So yeah, this is the main idea. Of course, well, as an idea, it's a simple idea. However, from a biology perspective, so this mechanism is very well studied. It's not that difficult to be implemented. This one, however, it's something, well, up to a point it has been studied. However, the secretion of pheromones to the environment upon the transcription being triggered is something that has not been done before. So it's pretty challenging from to build it. And so in case this fails, then we will use exogenously provided pheromones to test the system and the communication channel and the receiver. Okay, and then the project. The project objectives is to build the test bed and then to characterize the community. So to build a model for the transmitter, to build a mathematical model for the receiver, to build a model for the channel, and after we have the channel model, the physical channel model, as done in other test beds, to build appropriate modulation and coding schemes for to optimize the bit information exchange. Yeah, and the approach that we have adopted so far, because actually I have a control systems background, so the modeling procedure, apart from the diffusion part, we will follow dynamical differential equation state-space models, and there are, as I will show later, there is a rich literature on this specific system on how to model it. Of course, we have to complement those models with the new things that we will introduce into the system to make it happen. Okay, so this is I've talked about the communal. I've talked also about the mechanisms. So as I said, the yeast cells, they are haploid. They have two mating types. They secrete pheromones. Pheromone is a peptide. So I do expect it to be a bit slower than lighter particles. Now the benefit of peptides though is that they are sufficient, so it might be possible to carry more than 1 bits, to use them to carry more than 1 bits of information. Yeah, so the other thing is that the mating procedure, which we will be considering mutants, so they will not actually mate in the end. All right, so we will use the mechanism to secrete and receive pheromones, but they will not mate in the end. Now, the mating procedure, it lasts about 4 hours. However, as I will show later, it will not take 4 hours, because this would mean very slow bit rates, information exchange. Yeah, so, and actually it's pretty interesting. So this is the way, the steps that they follow, how they mate and stuff. And actually, so they secrete the pheromones, then they stop, they prepare for the mating procedure, so they can have this shape. Now, the reason that they have this shape here is that they built a pheromone concentration gradient for the one to be guided towards the other. And actually, we want to, in our analysis, we want to investigate how this concentration gradient forms and appropriately model it to investigate the transfer of the particles through diffusion via these gradients. Okay. Now, as I said, there are 2 types. So we will be taking. So we have the transmitter, and we have the receiver. We will be using the galactose pathway for the transmitter side, and then we will use the pheromone pathway for the receiver side. Now, even so, the transcription of genes, okay, for the conversion of galactose to glucose, it can be controlled. This is for the transmitter, based on the environment that you have. So there are 3 states. Let's say in a neutral environment using glycerol or raffinose, the GAL genes, they are not expressed. However, in the presence of galactose, they are expressed. In the presence of glucose, they are repressed due to the repressor protein, this MIG-1P. Now, why are these important? Now, even the mechanism with which this works, it's, there are different versions of it, so it's not well established, all right? So, however, this is one model, okay? So we have these, the GAL genes, so in the absence of galactose, the GAL 80 is attached to the GAL4, and so it does not allow it to start the transcription. In the presence of galactose, this GAL3 it attracts the GAL80, and then GAL4 is available it becomes available to start the transcription. And then once the transcription begins, then they will engineer the system through a particular mechanism to release pheromones into the medium. Now, as I said, this this mechanism by which was to release this has not been done before in the literature. So it is a novel from a biology perspective. There is novelty there in the mechanism. Now, as I said, this is a well-known, it's a well-studied system. So there are different models. Actually, this is one of the models. There are even more. Here, so this is a 5-dimensional state space equation, non-linear one. There are even more complicated 9 states. There could be more. I mean, we are still at the very early stages of reviewing all this stuff. But it does help to give us an indication of the timings that, the amount of time that these take to initiate the transcription. Now on the receiver diffusion model, this is very preliminary. Now for, as I said, we will be pursuing a more detailed analysis because we have to take into account the gradients, the concentration, pheromone concentration gradients, to build a more realistic model. Now, for the receiver side, so we have the G-protein couple receptors. It's this ST2 that you see over there. So upon the alpha pathway, the pheromones triggering the pathway, then they induce the release of this G-beta gamma, and then there are a series of steps leading to the expression of this ST12. And this ST12 here, it will be again engineered to express green fluorescent protein, which will be detected. And again, so there are many, many steps. And here it's an even more complicated model. Not complicated, I would say higher dimensional. So here we have, if I'm not mistaken, it's 35 states for it. But it's well established, and they are providing in the living systems biology. They provide even the initial conditions for. So yeah, just some initial. So we can see. So on the center side, if we simple MATLAB simulations using the state-based models, so we can see that this takes about 400 seconds to converge, and the other thing is that this is on the center side. Now here, once you, so this is, it turns it on. Now to turn it off, you need to, so to create bits, series of bits, so it's on, off. So you need to put, so here it's galactose triggered, then you have to put glucose to turn it off, galactose, and so on. Now there are established models for the repression as well. So this is, yeah, for the receiver response, surprisingly, it has been, so if you apply the pulse of pheromones, it's surprisingly fast. So I had a discussion with the biologist. They say that it might be the case, but of course, this is something that we need to verify using experimental setup. And of course, these are just, yeah, I don't know if you can see. So these are first outputs of the experimental setup. So this is without galactose. In the presence of galactose, you can see the green. At the beginning, when they sent me the slides, I was, so what did they send me? But then I noticed that there were some green spots there. So, yeah, so this is it. As I said, it's very preliminary work, but I thought it would be interesting to open up some discussions on this specific setup. Thank you.
Thank you very much.
Thank you.
Are there any questions?
Yeah.
First of all, thank you for the interesting idea. And when I'm looking at this topic from a communications perspective, I know this is a really early stage, but could maybe the color, the fluorescent color, the wavelengths be changed? Could it be engineered that it's not only green? Because at this point, I'm thinking of simple on-off keying.
Yeah.
Maybe we could encode bits also into the color of this fluorescent part.
Maybe, I mean, these are things that are, all these, this question, I need to, actually, what impresses me is the fact that how, the amount of stuff that biologists can engineer. So even because they don't have any idea about communication. So I mean the biologists. So what do you want? They said. So I want to have bits, and then when there is it goes down. So when it's off, I want the green line to go off. You want it to go off. So we need to create this kind of green fluorescent protein that dies away. If you wanted that to be maintained, it's a different green fluorescent protein that it maintains the intensity throughout the time. So I don't know the answer to that question, but it might be possible. That's what
It was just an idea.
Yeah,
also, another point would be to apply some different concentrations so that you have different intensities, for example, of the color. So you, for one time period, you could have a really bright green fluorescent.
Yeah, of course, in that case, because what you're trying to do, because at the end of the day, you need the green fluorescent protein to detect the amount of pheromones that have hit the receiving molecule. It needs a bit of thought on, so playing on, even the color. So when will it be red and when will it be green, let's say. Is it when it needs, but I think it could be done.
Great. So
Yeah.
maybe another quick question.
Thank you for a very great presentation. Now, my question is about this mating process that you talked about. Then you don't want to model this mating process. You're just
We model up to a particular point. We don't model the mating. We will be utilizing and modeling because the mating process it's in different stages. So not all the stages will be. So this will be ST mutants. So they will not undergo all the stages.
you just
So they will undergo up to a particular stage, and it is those stages that we will model.
Yes.
No, not the whole, because the whole thing takes 4 hours. You don't want to take 4 hours when you speak.
Your target is communication using molecules, and then you don't think about the final process that really cells do. Then you don't.
So what we want, the motivation is here. So we want to engineer this because different from what it exists in the literature. So we have engineer cells for transmitter, engineer cells for receiver, and the communication medium, which is it's diffusion based again, which is similar to. But you have all the elements in engineered cells. That's a bit different than what has been reported in the literature so far. And the other thing is that these yeast cells, they can do a lot of stuff with them. So we can engineer them there is a high degree of flexibility in what we can do with them from the biology perspective. And that's the main benefit. Of course, let me point out that so this is nice. The main thing, which still in many of these molecular communication studies, the main thing, the main question is, so what is the application? Okay, now this is nice from, let's say from a basic research approach, but what is the application? All right, so, and that's why I have invited Kostas to be part of this project. One potential application is yeast biosensors, which are I'm not sure if it's that hot, but it's an active area of research. So this is one potential application, except from the basic research side of such a project.
Can I ask another question?
Sure.
And
Maybe we can just leave this to the lunch afterwards.
Yeah.
So leave the discussions
Okay.
offline. So thank you again.
Thank you. Thanks.